Here's a question almost nobody asks themselves before breakfast. Does it matter what you eat first, or just what you eat? Most people picture blood sugar control as something that happens meal by meal, almost like a reset button gets pressed every time you sit down to eat. Eat well, glucose behaves. Eat poorly, it spikes. Simple, tidy, and, according to a growing body of research spanning more than 4 decades, not even close to the whole story. What actually happens is stranger and, frankly, more interesting. A handful of decisions made in the first 30 to 60 minutes after waking up appear to set a kind of metabolic tone that persists for hours, shaping how your body responds to food you haven't even eaten yet. Researchers first stumbled onto pieces of this pattern in the years right after World War I, lost track of it for decades, and then rediscovered it with modern tools that let them watch glucose rise and fall in real time. The pattern kept showing up in study after study, in different countries, in people with completely different diets, which is usually a sign that something biologically real is going on, not just a statistical fluke. By the end of this video, you'll understand exactly what that early pattern is, why a 1982 study out of Toronto changed how scientists think about breakfast entirely, and what your gut is quietly signaling to your pancreas before a single molecule of sugar ever reaches your bloodstream. There's also a wrinkle in this story that most people never hear about, a limit to how far this effect can be pushed, and I'll get to that later on. For now, let's start with the basics of what's actually happening inside you every time you eat. When carbohydrate-containing food lands in your stomach and small intestine, enzymes break it down into individual glucose molecules small enough to pass through the intestinal wall and into your bloodstream. This isn't instantaneous. Depending on what you ate, glucose can start appearing in blood within minutes or trickle in over a couple of hours, and that speed alone changes everything about what your body does next. The faster and more concentrated the glucose arrival, the sharper the spike your blood sugar sensors detect. Your pancreas is loaded with beta cells that function almost like glucose thermostats, constantly sampling the blood passing through them. The moment glucose concentration climbs, these cells release insulin, a hormone whose main job is to unlock cells throughout your muscles, liver, and fat tissue so glucose can move out of the blood and into storage or immediate use. Nobel-level biochemistry underlies this process. Insulin binding to its receptor triggers a cascade that physically shuttles glucose transporter proteins called GLUT to the cell surface, a mechanism described in detail by Alan Saltiel and C. Ronald Kahn in their landmark 2001 review published in Nature. Without this transporter movement, glucose would simply pile up in the bloodstream with nowhere to go. What emerges from this entire sequence is something researchers call a glycemic response, essentially a curve that rises after eating and falls back down as insulin does its job. Here's the part that surprises most people. 2 meals with identical calories and identical carbohydrate content can produce dramatically different curves, one steep and short, the other flatter and longer. The food itself only explains part of that difference. What explains the rest is where things start getting genuinely fascinating, and it has almost nothing to do with what's on your plate. In 2015, a research team led by Alpana Shukla at Weill Cornell Medical College published a small but striking study in the journal Diabetes Care. They took people with obesity and type 2 diabetes and had them eat the exact same meal, protein, vegetables, and carbohydrate, on separate occasions, changing only the order in which the components were eaten. When carbohydrate was eaten last, after the protein and vegetables, postprandial glucose was measured substantially lower when carbohydrate came last instead of first. Insulin requirements dropped substantially as well. Same food, same calories, same person, just a different sequence, and the entire glucose curve changed shape. So why would sequence alone change a hormonal response this much? Part of it comes down to gastric emptying. Fiber and protein physically slow the rate at which the stomach releases its contents into the small intestine, which spreads glucose absorption out over more time instead of dumping it all at once. But there's a second mechanism at work here too, something happening at the hormonal level before glucose even reaches your bloodstream in large amounts. And it's a piece of physiology most people have never heard of. That part of the story is coming up shortly because it connects directly to what your gut is doing in those first few minutes after you start eating. In 1982, David Jenkins and colleagues at the University of Toronto published a paper that quietly reshaped how researchers thought about meal timing. Jenkins, already known as the scientist who developed the glycemic index, discovered something unexpected while studying how the body handles sequential meals. The first meal of the day appeared to influence how well the body handled the second meal hours later. He named it the second meal effect, and it turned out to be one of those findings that seemed almost too simple to be true, yet replicated again and again. Nearly 3 decades later, in 2009, researchers Anna Jovanovic, Jean Girard, and Roy Taylor at Newcastle University tested whether this effect held up in people with type 2 diabetes, a population where many normal metabolic responses break down. They compared lunchtime glucose response on days when breakfast had been eaten versus skipped entirely. The results were dramatic. The rise in blood glucose after lunch was roughly 95% smaller on days when breakfast had been consumed first. The same research group followed up in 2010 with an even more targeted experiment, giving participants a small high-protein snack before breakfast, and finding it reduced the post-breakfast glucose spike by nearly 40%. What explains this carryover effect from one meal to the next? The Newcastle team traced part of it to free fatty acids circulating in the blood. When breakfast is skipped, fatty acid levels climb higher by lunchtime, and those elevated fatty acids directly correlated with a bigger glucose rise after the meal that followed. Eating earlier in the day appears to suppress this fatty acid buildup, leaving the body better primed, more sensitive to insulin, less metabolically defensive, for whatever comes next. In other words, breakfast isn't just fuel for the morning. It's information your body uses to calibrate its response to lunch, and possibly dinner, hours before either meal happens. Back in the 1970s, a German physiologist named Werner Creutzfeldt noticed something that didn't fit the standard model of insulin release. When people drank a glucose solution, their bodies produced 2 to 3 times more insulin than when the exact same amount of glucose was infused directly into a vein. Same sugar, same final blood glucose level, yet wildly different insulin output. Something about eating, separate from the glucose itself, was telling the pancreas to respond more aggressively. That something turned out to be a pair of hormones released by cells lining the small intestine the moment food passes through. GLP-1 and GIP, collectively known as incretins. As Michael Nock and colleagues have documented across decades of research, these gut hormones travel through the bloodstream to the pancreas and essentially prime the beta cells, amplifying how much insulin gets released in response to the glucose that's about to arrive. This is the missing piece connecting back to food order. Vegetables and protein eaten before carbohydrate appear to stimulate stronger GLP-1 release, which helps explain why the sequence of a meal changes its glucose impact even when total calories stay identical. There's a third layer to this story, one tied to something far older than any of the research covered so far. In 1919, 2 researchers named Louis Hammond and Isaac Hirschman noticed something odd while studying blood sugar at Johns Hopkins Hospital. When they gave a person glucose more than once in the same day, the body handled the second dose noticeably better than the first. 2 other physiologists, Hans Staub in 1921 and Carl-Traugott in 1922, independently confirmed the pattern in healthy subjects, and it eventually took both their names, the Staub-Traugott effect, Give someone a glucose load, wait a couple of hours, give them another identical load, and the blood sugar rise the second time comes down faster and lower, even though nothing about the glucose itself has changed. The effect has been retested with modern tools more than once, most notably by Breno Astiaraga, Jens Holst, and L.A. Ferranini's group in a 2009 paper published in the American Journal of Physiology. They gave 17 healthy volunteers 2 oral glucose tolerance tests, spaced 3 hours apart, and found measurably improved beta cell function on the second test. The pancreas responded more efficiently the second time around. What's refreshingly honest about that paper is that the authors openly admit the underlying mechanism still isn't fully settled. Leading theories point to insulin's own priming effect on beta cells and to the first glucose load physically slowing the gut's processing of the second. But a century after the phenomenon was first described, nobody has closed the case completely. Here's why this matters for the bigger picture. The Staub-Traugott effect is a distinct phenomenon from the second meal effect Jenkins described in 1982. Jenkins was looking at how meal composition, fiber content specifically, carries over to influence the next meal. Staub and Traugott were looking at something more basic, repeated glucose exposure itself, independent of fiber, independent of protein, independent of anything except timing. Two separate research traditions, decades apart, converging on the same underlying truth, that your body doesn't treat each meal as an isolated event. It keeps a kind of running tally, and that tally appears to be shaped, at least in part, by what time of day it's being kept. There's another mechanism worth adding here, one that has nothing to do with glucose directly. In a study published in the British Journal of Nutrition, researchers Kirsty Evans, Pirjo Kusala, Maria Cruz, and Keith Frayne tracked what happens to fat from an earlier meal when a second meal arrives. Dietary fat gets packaged into particles called chylomicrons and released into the bloodstream, and remnants of those particles can still be circulating when the next meal shows up. The composition of that second meal changes how quickly those leftover fat particles get cleared, which matters because circulating fatty acids are one of the factors that interfere with how efficiently muscle and liver tissue respond to insulin. In other words, your body is managing fat carryover and glucose carryover simultaneously, and the two systems talk to each other more than most people realize. It's worth being precise about how far this carries, though, because overstating it would misrepresent the science. The strongest, most consistently replicated evidence covers the window from breakfast to lunch, a handful of hours. Evidence extending meaningfully into dinner is thinner and more mixed, partly because so many other variables pile up by evening. Physical activity, stress, what was eaten at lunch, how much time has passed since waking. So the honest way to describe this is a fading gradient rather than a flat effect, lasting equally all day. Strongest in the first several hours, present but harder to isolate later on. So here's a question worth sitting with before we go further. If your body really is keeping a running tally of everything you eat starting from the first bite, what does that change about how you think of breakfast? Not as a separate meal, but as a kind of instruction your body carries forward? Drop your thoughts in the comments. I read every single one. None of what's been described so far happens in a vacuum. Your body also runs on an internal circadian clock, and glucose metabolism is one of the systems most tightly wired to it. In 1991, Eve Van Cauter and colleagues at the University of Chicago, publishing in the Journal of Clinical Investigation, tracked glucose and insulin secretion across full sleep-wake cycles and found clear rhythmic patterns independent of food intake. Glucose and insulin secretion rose during nocturnal sleep and settled back to baseline by morning. And when subjects were kept awake through their usual sleep period, those same rises appeared again, timed too when sleep normally would have started, rather than to anything they ate. A more recent study pushed this further. In 2015, Christopher Morris and colleagues published a paper in the Proceedings of the National Academy of Sciences using what's called a forced desynchrony protocol, a method that separates a person's internal circadian clock from their actual sleep-wake schedule entirely. They found that circadian phase affected glucose tolerance through mechanisms separate from sleep itself and separate from how long someone had been fasting. Reviews summarizing this line of work consistently describe the same broad pattern. In metabolically healthy people, insulin sensitivity and beta cell responsiveness tend to peak in the morning and gradually decline through the afternoon and evening. If all of this machinery is primed to work best in the morning, what happens when breakfast simply doesn't occur? In 2005, Hamid Farshchi, Moira Taylor, and Ian MacDonald at the University of Nottingham ran a controlled crossover study in 10 healthy, lean women, publishing their results in the American Journal of Clinical Nutrition. Each woman spent 2 weeks eating a structured breakfast and 2 separate weeks omitting it entirely, while otherwise eating freely. The researchers found that the breakfast omission weeks were associated with impaired postprandial insulin sensitivity and a worse fasting lipid profile compared with the breakfast weeks, and total daily energy intake also tended to run higher when breakfast was skipped. A different and much larger study looked at this question from a longitudinal angle instead. Kylie Smith and colleagues, publishing in 2010 in the same journal, followed participants from the Childhood Determinants of Adult Health study over several years and found that habitual breakfast skipping was associated with worse cardiometabolic risk factors later in life, including markers related to insulin resistance. It's important to be precise about what this second study can and can't tell us. It's observational, tracking people over time rather than assigning them to skip or eat breakfast, which means it shows an association between the habit and the outcomes, not proof that skipping breakfast directly causes them. People who skip breakfast regularly may differ from those who don't in a dozen other ways that also affect metabolic health. None of this adds up to a closed case, and it's worth being upfront about exactly where the uncertainty sits. Most of the food order and meal sequencing studies covered earlier involve somewhere between 15 and 25 participants, tracked over a single day or a few weeks at most. The long-term data showing sustained glycemic improvement, Kajiyama's group followed patients for 2.5 years, comes almost entirely from people already diagnosed with type 2 diabetes. Whether the same magnitude of long-term benefit shows up in metabolically healthy people who make this a daily habit for years hasn't been tested with the same rigor. The mechanism question hasn't fully closed either. Even the Staub-Traugott effect, confirmed and reconfirmed for over a century, still doesn't have a fully agreed-upon explanation, as Farinini's own research group acknowledged in their 2009 paper. When a phenomenon this well replicated still has open mechanistic questions, it's a reasonable signal that the full biological picture involves more moving parts than any single study can capture. There's also the question of individual variability that researchers are only beginning to unpack. Some work on chronotype, whether someone is naturally more of a morning or evening person, suggests that the ideal timing of meals relative to the body's internal clock might differ from one individual to the next, meaning a pattern that works well for one person's biology might work differently for someone whose internal rhythm runs on a different schedule. And there's a confounding problem baked into almost all of this research. People who naturally gravitate towards structured, protein-forward breakfasts often differ from people who don't, in plenty of other ways too. Sleep habits, activity levels, overall diet quality, any of which could be doing some of the work credited here to meal order and timing alone. Pull every thread from this video together, and what emerges isn't one single trick, despite how tempting it is to look for one. It's a layered system. The order in which food is eaten changes gastric emptying and incretin release. The first meal of the day primes the pancreas through a mechanism first observed over a century ago. Circadian rhythms tilt the entire system toward peak efficiency in the morning hours. And skipping that first meal appears to forfeit all of it at once, based on both controlled and long-term observational research. What's genuinely remarkable is how independently these findings arrived. Jenkins wasn't thinking about circadian biology in 1982. Van Cauter wasn't thinking about food order in 1991. Staub and Traugott, working in the 1920s, had no concept of incretin hormones at all. GLP-1 wouldn't be identified for another 6 decades. Yet each research thread, approached from a completely different angle, kept landing on the same underlying truth. Your body treats the start of the day as a kind of calibration point, and what happens in that window echoes forward for hours. Where the science still needs to catch up is in tying all of this together in long-term, well-powered studies in healthy populations. Something that, as of now, simply doesn't exist at the scale this topic deserves. What does exist is a remarkably consistent signal across nearly 100 years of research, coming from completely different scientific traditions, all pointing at the same handful of hours after you wake up. Whether that signal changes anything about how you personally think about your mornings is worth sitting with. But the data behind it is about as solid as this kind of research gets. If this changed the way you think about your morning routine, hit like. Subscribe for more videos exactly like this one, and share it with someone who could use a science-based look at what's really happening in their body every day.